Good morning, good afternoon, and good evening from Japan. My name is Kazuyo Moro from Osaka University and Riken IMS, and it's my great pleasure to welcome you to Global IMMUNOTALK 2023. Today, before moving to today's talk, I would like to give two reminder. Next week's speaker is Kataya Simon from University of Oxford. So please don't miss it. And the another announcement is about how to make a question. So in this global IMMUNOTALK, you cannot directly ask a question to the speaker, but you can make a question through the Twitter. So I'm gonna make an announcement about how to use a Twitter for the question after the talk. Okay. So now it's my great pleasure to introduce Jim DeSant from Institute Pasteur for today's speaker. So he received a combined MD PhD from Cornell Medical College and the Sloan Kettering Institute in New York City, and finished the postdoctoral training with Alain Fischer in Paris and Klaus Lajowski in Germany before creating his own lab in 1999 in the Institute Pasteur. So he currently holds the position of professor in immunology department at Institute Pasteur and director of research with the French Medical Research Institute. So with more than 30 years of expertise in fundamental and translational immunology, his main scientific interests are in the area of lymphocyte biology and cytokine and transcription factors and signaling pathway in the development and function of both adoptive and innate lymphoid cell in mice and also human. So in parallel, over the last 20 years, he has developed a series of humanized mouse model for the immune system that allows him to prove the fundamental question in human immunology, especially in the relations to the infectious disease. Most recently, he has developed an integrated systems immunology approach using nasal swab samples to assess the biology of the active mucosal site in healthy individuals and patients suffering from the respiratory diseases. So I'm sure many of you joining to today's talk know that his excellent work on ILC3s, and his talk title is Innate Lymphoid Cell Development, Differentiation, and Dynamics. So I expect that we will probably hear some very interesting story about ILCs today. So Jim, if you ready, you have already turned on your camera. So thank you very much for joining to this Global IMMUNOTALK. It's my great pleasure to introduce you. And maybe as you know, in this program, we start with one question for the speaker. And today's question for the speaker is, who is or who has been a scientific role model for you? And also, I want to know the reason why you choose that person. So could you answer to this question?
Sure. Thanks, Kazuo, very much for the introduction, and thanks for being here live. It's I know it's late in Japan. So I've been lucky because there have been a couple of different individuals that have been mentors to me over the years. Neil Flamenberg when I was a PhD student, and as you mentioned, Alan Fisher during the postdoctoral period, Klaus Drzeski when I was an EMBO fellow in his lab in Cologne. And all three of them were very supportive and incredibly strong mentors in the way they were able to support me and show me ways to do science. But I think one of the key points about each of these individuals and another person in particular, which I'll come to in a minute, is that they gave me freedom to do what I wanted to do. So that was really important, because often you come up with new ideas and you want to move in new directions, and if you're stuck, you can't do that. But if you're free, you can. And I think the person who's probably influenced me the most in that respect is Max Cooper. Max Cooper is really a prototype of a physician scientist in the best degree, and he has an amazing breadth of knowledge, and every interaction with him was always a pleasure, because he's extremely curious, and he was also always ready to ask questions and to think about the problem that you were doing, and it wasn't about him, it was about you. And that's someone I think that really influenced me, because it showed me how basically you can be intense and you can be motivated, but you can also be extremely generous and curious and helping others. And Max was really incarnating all those things. And that's why I think I would put him at the top of the people who've influenced me by the way he acts and by setting that role model. He's really a class act.
Thank you very much. Yeah, so I hope you are now like someone in your lab thinks so as for you, and you're helping your lab member well, and I hope someday that guy will answer your name in this question. Okay, so if you're ready, please show us your slide and let's start your talk.
Okay. So is that visible?
Yes.
Perfect. So again, thanks very much to all the organizers, especially because for this great opportunity to be part of Global IMMUNOTALKs. And I'm looking forward to sharing this data that has been generated in the lab by several individuals over the past couple of years. And again, it's centering around this population of innate lymphoid cells and how they're basically still being in some ways discovered, but clearly important players in many aspects of immune responses. So it's really interesting to see the symmetry in science, and the lymphocyte subsets also show this beautiful symmetry at the level of the innate and adaptive immune system. So we know for many years about adaptive T cell diversification, including both CD4 and CD8 subsets, and the helper subsets that are defined within the CD4 T cell compartment. And these are characterized by different profiles of cytokine production and by transcription factors that help generate those phenotypes. And we've known for quite some time as well that the CD8s are a different type of cell, having much more activity in terms of killing targets. And that's due to, again, a transcription factor expression profile that generates these granules that are important for killing target cells under different contexts, including inflammation and stress and in cancer. Now, we've had an innate homologue of the CD8 cytotoxic T cell for a long time. We've known about these cells. These are natural killer cells, and we've known about them for over 50 years. But it's more recent that we've discovered that there are also subsets of innate lymphocytes that mirror the diversity in the helper T cell compartment. And these are now denoted innate lymphoid cells, and there are three main types that are shown on this slide that mirror the Th1, Th2, and Th17, 22 T cells. So they have an amazing similarity in terms of the transcription factors that drive cytokine production from these cells. But unlike T cells, they don't have T cell receptors. So this, of course, begs a number of questions, amongst them why we need this kind of diversification and why we need to have cells which bear antigen receptors or don't bear antigen receptors producing these cytokines within our bodies. So these cells have been the focus of studies for about the past 15 years. And we've learned a lot thinking about them in the context of infection. It's clear they're not cells that act on their own. These are cells that require signals from the environment to become activated and to participate in immune responses. And as we know, there are different types of immune responses that can be categorized, and these are generalized ways to think about immune responses, of course. Type 1, type 2, type 3, or Th17 type responses are well characterized for T cells, and analogously, NK cells and ILCs also fall into these categories as shown on the slide. So these cells are not directly pathogen sensors, although they have some sensing receptors on their surface. They do largely depend on signals that are elaborated within the environment that are produced by sentinel cells. So sentinel cells can spew out cytokines that can then activate NK cells or ILCs, and they will then convert them into new types of factors that are important for immune responses. And I won't go in depth through this type of a slide. You understand that the production of interferon gamma is important for activating macrophages, and type 2 cytokines are important in the context of parasitic infections. But these cells are not only involved in infection protection, they're also involved in many other aspects of tissue homeostasis and health in the organism. And that's become more and more clear as we start to analyze these cells, their receptors, and what can activate them. And we've learned that ILCs can go beyond infection. They're important in many systemic regulatory mechanisms. Metabolism, for instance, ILCs play a very important role in the balance between energy metabolism and fat homeostasis. And the different types of cells can either have positive or negative effects within this context. We know that ILCs also produce many factors that are important in tissue repair. So the idea that these cells might be critical for rebuilding tissues after stresses and after infection has come into the fold. And most recently, a very intricate and important interaction between the nervous system and ILCs has been demonstrated with a multitude of different types of neurotransmitters and nervous system signalers that are present and effective in terms of modulating NK and ILC biology. And again, these are in both positive and negative ways, suggesting that a balance between these signals is achieved in vivo, and that they can be also modified and perturbed by different types of challenges, either infection or disease. So we were interested initially from the standpoint of how these cells developed. And we were lucky to have some support through the European community for a long-term grant, which allowed us to explore some of these different aspects of the biology of NK and ILCs shown on this slide. And we initially focused on how they develop. And the dogma at the time was that these are probably produced in the bone marrow, and the mature cells then are able to go through the circulation and take up residence in the different end organs where they exert their functions. And using that model, of course, there had been already quite a lot of knowledge in terms of the way different lymphocytes are generated, NK cells in particular, and we've been studying that over many years. And it was very quickly adapted, the same kind of an approach to understanding how ILCs are generated. And many groups were involved in this work. And this slide from a review from Nicola Serafini in the laboratory give some idea of the complexity of this developmental pathway. And we learned a lot about the different signals that are involved in generating these mature ILCs that have been described initially from the lymphoid precursors that we know are important for generating both innate and adaptive lymphocytes. Now, along the way, key intermediates were identified, and some of the work that was generated from several laboratories, including Avinash Bandula and Andreas Diffenbach and Leidabert Bendelak, identified ILC precursors, which were critical intermediates in this pathway. And they were important because they were able to generate all of the mature cells, but not any other hematopoietic lineages. So they're really a key stepping stone to this developmental maturation pathway. And we wanted to understand that a little bit better. Our approach involved making reporters for these different transcription factors and then multiplexing them using different types of fluorescent reporters to look at then co-expression of these transcription factors in these intermediates. And in that type of approach, we were able, and when I say we, I'm talking about Wei Zhu and Dylan Cherrier, were able to show that these precursors in the mice are basically able to show developmental potential for all the ILC subsets, but in fact, they're not really hardwired in a way that you can see every step along the way, as we have shown in that previous slide. More, they're in a state where they can generate these different mature subsets, but they require environmental signals to be educated and to be differentiated along the proper pathway. So there isn't a strict pattern of differentiation, but rather a more flexible pattern of opportunities that these cells have. And depending on their environmental signals, then they can differentiate fully into the mature subsets that we know. Now, if we take that analogy and apply it to humans, we would think that these cells would be in the bone marrow in the humans, and we were thinking about going along that line and trying to identify those cells, but instead we started looking at these cells in the blood because that was a tissue and a sample that was much more easy to obtain. And Ai-Ying Lim in the laboratory made a very surprising observation when she was characterizing ILCs in the blood of humans, and that she found a population of cells that appeared to have this precursor potential, in that those cells, when purified and put into in vitro or in vivo assays, were able to generate all the different ILC subsets, but not any other hematopoietic progeny. And she went on to characterize those cells and look at them transcriptionally and look at their signatures. And it was clear they resembled very well the kinds of cells that we had studied in the mouse and been described in the mouse. And the example of the transcription factors that they express is shown in the bottom of the slide on the left. But clearly, again, they were not committed to one particular ILC pathway. They rather depended on signals to generate then this diverse subsets, depending on the types of signals they would receive. And this led us to propose a new way of thinking about how these cells develop, that they wouldn't be generated as a hardwired population in the bone marrow, for instance, but rather that the cells could actually be generated anywhere from a precursor that was not committed, but rather waiting to be educated in a particular context. So these human ILC precursors that we find in circulation would be able to differentiate in situ into the proper type of ILC that would be needed in that tissue, depending on the particular disease state. So Ai-Ying was able to show that there were signals that allowed these cells to expand and to differentiate. One of them was IL-1 beta, and that's also interesting because that's clearly activated in the context of inflammation. So a precursor that comes into a tissue, an inflamed tissue, then would expand, receive signals from the environment, and differentiate into a mature ILC subset, and that would basically depend on what kind of signals it would receive during that process in the tissue. So this concept is now supported by a number of studies as well in mice, and we use it to try to understand how the signals that are important for differentiating ILCs are basically generated and regulate this process. So it's interesting when you start to then compare this idea of differentiation of ILCs and differentiation of T cells, because the parallels turn out to be rather striking. Now, we know T cells, of course, develop into helper subsets in secondary lymphoid tissues under the influence of dendritic cell signals and antigen MHC complexes, which trigger the T cell. And that then generates the whole panoply of T helper subsets, depending on the context of that delivery of antigen MHC to the naive T cell. We would propose that in the ILC world, the naive ILC would be this ILC precursor, which then would have a similar type of differentiation pathway in tissues, depending on the types of signals that it receives. So there may be a trigger that then is similar to what would be the TCR-like trigger for an ILC precursor, and that in context of cytokines and other stimulatory molecules would then generate the differentiation process. So this idea about parallels between T cell and ILC differentiation are rather striking, and I'll show another set of examples of this in the effector stages in the latter part of the talk. So what evidence do we have that these ILCs that are in the blood are actually equivalent to naive ILCs? Well, phenotypically they are. They really resemble what would be considered as a naive cell. They don't proliferate. They're mostly CD45RA, which is naive marker. They express some L-selectin, so they may be able to get into lymph nodes as well as other tissues, but there's a large majority of them that are negative for CD62L. And they're not activated by any means. They're CD69 negative, and they lack all of these other markers that we associate with the mature cell that's differentiated. And furthermore, if we look at their metabolic profiles, it's very clear that they're not a highly activated cell in terms of metabolism. If you look at the different aspects of metabolic pathways, including glycolysis or fatty acid metabolism or the TCA cycle, you can see very clearly on this slide that the ILCPs are mostly dark, whereas NK cells and ILC2s have these pathways activated. And furthermore, on the right, if you look at mitochondria, which is, of course, very important metabolic source for dividing cells, activated cells, you can see that ILCPs have very poorly developed mitochondria, both in mass and in their potential. And this work from Laura Sarace, who was in the laboratory and now has her own lab in University of Bonn. So we have this idea about a T cell perspective for ILC differentiation. This is the model we're working on currently, trying to understand what could be the signals that are triggering these cells to differentiate into mature ILC subsets. The types of parallels, again, between ILCs and T cells during this type of process are pretty striking. So the naive ILCs, like naive T cells, have a lot of IL-7 receptor. When they're activated, that gets lost, the IL-2 receptor gets put up, and then IL-2 can drive the proliferation of those cells. This is exactly what happens in T cells when they get differentiated. So the big black box is, what are the signals that are allowing then the differentiation to these mature subsets? And that, of course, is an area of active study. So I'll shift now to the periphery. And this will be, from now on till the end, mostly a story in mice. And the mouse is a great model for studying, especially ILC function, because we can, of course, expose mice to different types of triggers and then understand how the immune response is generated in a longitudinal fashion, and also the diversity of the players that are present can be analyzed in depth. And if we think about the barrier in the intestine, it's of course a very complex interface. On this slide, you can see the immune side, which is behind the epithelial border. But of course, there is a very complex microbiome that's on the other side of this epithelial border, which is playing a very educational role in the way the tissue is evolving over time and the immune system with it. And we know many of the different players that are important in sampling these communities and detecting pathogens. We know that both T cells and B cells are generated specific responses, and IgA is also an extremely important mechanism for maintenance of this commensal host interface. But the different cells that are involved are including T cells and different types of T cell subsets and ILCs. So we know that there are many different cells involved, but these static images that we have of these interactions doesn't really help us to try to understand how it works. And sometimes we need to actually look at this directly. And that was the approach that we decided to take in the laboratory to directly assess the behavior and the dynamics of the ILCs within this compartment. And we were very lucky to have a collaborator very close nearby on the floor here in the building, the Metabolic building, and that's Philippe Bousseau, who's an expert in intravital microscopy, and he's been interested in many aspects of immune responses using this approach in mice. And Sasha Garcia in his laboratory teamed up with Nicola Serafini and Angelique Gerard in my lab to do the work that I'm going to present to you now. And basically, the question was, what do these ILC3s do in the gut in terms of their behavior, and how do they function in the context of different types of triggers, either under steady state or following inflammation? And of course, we knew nothing about this because there were basically no publications, although there was one paper from Fiona Parry's group that looked at some of the ILC dynamics in the crypts, in these cryptopatch structures. But there really wasn't a lot of data in terms of the way the ILCs were moving in the lamina propria or in the villus. So this was interesting to try to image and to try to understand the biology of these cells. So first, just some information on heterogeneity of ILC3s in the mouse intestine. So we know from a number of studies that there are several types of ILCs that are present in the intestinal barrier, and that includes both NKp46 or NCR positive ILC3s that are present and enriched in the villus. So these sit in the lamina propria underneath the epithelial border, and clusters of cells that are including both dendritic cells as well as ILCs that are of the lymphoid tissue inducer cell type that sit at the base, and they're shown on the right bottom part of the slide as those blue clusters that are at the base, and then the single NKp46 positive ILC3s are in the villus, and they're more sparsely populating that compartment. So we wanted to look at both of these cell types, and we had to find ways to image them. And we were fortunate, again, to have some interesting mouse strains that could help us. And one was from Gerard Eberl here at the Institute, where the ROR gamma locus was tagged with GFP. And a second was a IL-22 tomato reporter that was made from Scott Durham at the NIH that together allowed us to focus in on a very small subset of cells, which were ILC3 or Th17/Th22 oriented T cells. So these reporters were well expressed. They were easily imaged using this approach. Easily, I say that with, again, a caveat. Imaging in the intestine in a live mouse is very challenging. And the work that Sasha and Angelique and Nicola had to basically work to generate these images is really an amazing tour de force. So we have these reporters. We needed a way to have them specifically in ILCs. That was done by generating a chimeric mouse where the reporter was placed on a RAG background. This then allowed you to isolate the ILCs from any other T cells that could be expressing IL-22. And those were mixed with B6, actin, cyan, FP cells to generate then these mixed chimeras that could be imaged. So here are the images. And here we're looking first at the small intestine in the villus in these chimeras. And the blue cells include T cells, B cells from the B6 donor, and the green or green-yellow cells are the ILC3s. And what you can see is that the blue cells, a fraction of them are moving quite a lot. And surprisingly, the green cells are not really moving much at all. So this is in the villus. And we also took advantage of the fact that the crypts could be imaged in these mice. So here are the crypts with the same reporter, chimeric reporters. And here you can see again that those cells that are green or green-yellow are basically immobile. So they're not really moving at all. Whereas there are blue cells that are outside the crypts and a few that are inside the crypts that are quite mobile. So this can be quantitated, and it's important to quantitate this. So if you look on the blue cells, basically a fraction of them, which represent the T cells, have an elevated mean velocity. So they're moving quite easily. And those are not arrested. So they're not fixed in space and time and space. In contrast, if you look at the GFP positive cells, either in the villus or in the crypts, they're very different. So most of them are arrested, and the ones that move have a reduced velocity. So they're not moving nearly as much as the T cells. So this was a bit of a surprise to us because we imagine that a cell that's involved in maintaining a border might actually be wanting to move around and look at the different parts of the border, but in fact, it's not the case, at least with this type of system. So we decided to go with another system to make sure that it wasn't an artifact. This was another reporter mouse, which included the NKP46 GFP mouse, and again, the same 22 reporter. Now, only one cell can be double positive in this particular type of mouse, and that's the villus NKP46 positive ILC3. And that can be imaged as a green-red cell. So here's the image. So the cells are tracked, and there's little boxes around them so you can get an idea about which cells are the relevant ones. Clearly, the other green cells that you see here are likely to be NK cells that are moving around quite a bit. But in contrast, the ILC3s again have a low mean velocity and they're mostly arrested. So again, in two separate systems, the ILC3s that are in the intestine are basically very poorly motile cells. Does that mean that they can never move? Well, we tested that by activating the system, and the system that was used was to inject a TOL5 agonist, flagellin, into the mice, and that creates an acute inflammation. And under this circumstance, now you can see as you image these cells that they're not fixed. They can move around. It's just that under normal circumstances, something's restraining them. So here you can see clearly in the bottom that the cells that are activated under inflammatory conditions have a much higher mean velocity, and their arrest coefficients are strongly reduced. So they displace and they patrol inside of those villi. So summarizing what I just showed you, there clearly are several states that tissues can take, and in the context of those different states, the ILCs can have different behaviors. Under the steady state, they're mostly immotile. They're in a position where they are not really exploring their environment. But if they're activated, and that activation in the context of inflammation allows them then to take up a patrolling behavior, explore their environment, and presumably interact with other cell types, including the epithelial border. So we were curious to understand what actually restricts these cells during the steady state, because normally, like we imagine, these cells are active in terms of protecting the barrier and containing commensals. How is it that they're not able to explore that space in normal conditions? So we thought about the different ways in which these cells might be restrained, and one of the obvious things to test were other types of lymphocytes that might be important in this balance between patrolling and non-motile behaviors that we see. So we simply went back and imaged the RAG2 deficient reporter mice that we had in the laboratory. And here you can already see, before I show the movies, that there's clearly some differences now in the number of these cells, and as well in terms of the activation of state of those cells, as many more of these cells are yellow, meaning they're expressing IL-22. In addition to the numbers and their activation profiles, these cells also clearly have a different behavior at steady state. So these cells are very motile and exploring the space in the villus. And again, this can be quantitated and is clearly much higher than what we saw in the chimeric system or in the NCR GFP IL-22 tomato mouse. RAG mice differ from conventional mice by adaptive lymphocytes. This can include T cells or B cells. So a very straightforward experiment was to put back T cells and assess the behavior of those motile ILCs that we see in the RAG mouse. So this was done by adoptive transfer. And a few weeks after the adoptive transfer, you can see that the villus is now fully populated with donor T cells. And that has a dramatic effect on the behavior of those ILCs in those compartments. So you can see now, again, imaging these red cells within these mice that have received T cells, that the ILC patrolling activity in the RAG mouse is now basically abolished. T cells are able to suppress that patrolling behavior when they're transferred into these immunodeficient reporter mice. So how are T cells doing this? We thought about interactions between different types of adhesion molecules and different types of soluble factors. And we know that chemokines and chemokine receptors play an important role in regulating both migration and activity within tissues. So we looked at the different chemokine receptors that were expressed on ILC3s, on T cells, and that analysis led to a candidate, which was CCR9. CCR9 is the receptor for CCL25, produced by different cell types within the intestinal system inside the barrier. The expression levels of CCR9 were interesting in that they were highly expressed on T cells. This receptor also well expressed on the NKp46 villus ILC3s, but not any noticeable expression on the CCR6 positive ILC3s that are in the crypts and the ILFs. So this matched pretty well with the types of interactions that we were seeing when T cells were transferred in to these RAG mice. And furthermore, the ligand was regulated in a very interesting way, in that the ligand was upregulated in RAG mice, where we saw patrolling, and that upregulation was modified when T cells were transferred into those immunodeficient RAG mice. So clearly the pattern of ILC3 behavior and patrolling and the expression of the CCR9 ligand were very closely paralleled. So we first looked at whether CCL25 was important for the patrolling. So this used neutralizing antibodies, which are injected into the mice, and then the ILC3s were imaged. So this, you can see the movement of these cells in steady state. After injection of a neutralizing CCL25, the cells start to slow down after an hour. And by 2 hours, basically, the cells have stopped moving. So the displacement graphs and the velocities and arrest coefficients are all shown and quantitated in the bottom of the slide. But I think the images speak pretty well for themselves. This is clearly an important factor that regulates ILC3 patrolling. Now, it's interesting that the receptor CCR9 is upregulated on gut homing T cells during their differentiation in draining lymph nodes. And that leads to a model where these cells might go back into the villus and interact and interfere then with that interaction of CCL25 with the ILC3. So this was easily tested using this adoptive transfer approach that I showed you earlier. You simply have to compare then the ability of CCR9 competent or CCR9 incompetent T cells to affect the patrolling activity of ILCs. So the idea is that the CCR9 positive T cells would interfere and that CCR9 negative might not. And this turned out to be the case. So again, the data is shown in the bottom of the slide. You can see that when CCR9 positive T cells are injected into these rag mice, the ILC3s are inhibited, their patrolling activity is reduced, and they become arrested. Whereas when you transfer in the CCR9 negative T cells, now the ILC3s that are present in those villus compartments are still motile, they still displace, and they basically have wild type behavior. So the T cells and the CCR9 positive ILC3s in the villus are basically competing in some ways for this ligand. So what is that actually important for is a good question. And what is this patrolling activity doing in terms of interactions with the border? So this was studied by many groups trying to understand the role for ILC3s in the maintenance of the barrier, the epithelial barrier. And it's been shown that 22 is an activator of epithelial function as well as production of antimicrobial peptides. So it's possible that this patrolling behavior is actually influencing then the barrier and allowing it to be more effective in terms of containing commensals and also promoting epithelial health. So I won't show you all the data. It clearly has a role in the barrier function. If looking at the fluidity of the barrier, it's clearly defective when this patrolling behavior is inhibited by antibodies against CCL25. But this is another important point in that when that patrolling behavior is neutralized by antibody, then the epithelial border starts to have defects in terms of homeostasis and the epithelial cells show increased apoptosis. So maintenance of the barrier by ILC patrolling in this context is important, and it's presumably, and we'll show you the data, supported by the fact that these cells are making IL-22, and that's allowing then the epithelial cells to have a robust function. So for this part of the talk, I think we've added some new knowledge in the area of how ILC3s are operative in their function at barrier sites in the intestine, but maybe elsewhere. And it's interesting to look at this in the context of how the immune system develops with time. And earlier studies in newborns showed that, in fact, the ILCs that are present in the intestine are highly active in this period. And it's when the microbes and the weaning of those newborns takes place, that you start to see changes in the composition of cells within the lamina propria. And one of the major changes that occurs during this period is an influx of T cells. And that influx of T cells has a dramatic effect on the activation of ILCs. And that was shown in an earlier paper from Yasmin Belkade that was published in Nature. We find that this T cell is active throughout life in terms of its ability to modify ILC3 function. And here we're looking at the behavior and the activation state of those cells. But that's not an irreversible change. Those ILCs can still be called into action depending on the inflammatory context. And when the system is activated by infection or by other types of challenges, then those resting ILCs can come out of their non-motile state. They become more activated, and they can generate then signals that are important for immune defense, presumably through secretion of IL-22 at the epithelial surface. So this is followed now by the last bit of the talk, which is another story addressing the question about ILC3 adaptation and the biology of these cells. And again, this is a parallel that we wanted to take with how T cells are generated and are functionally differentiating in the context of repeated stimulation. And we know that a T cell becomes activated and enters into a memory phase following encounter with antigen. And that memory cell has specific properties that allow it to be able to respond more rapidly and more robustly when it's recalled by the same antigen MHC complex later in life. And we were curious to know if that type of biology, in terms of adaptation to antigen counter or equivalent activation, would operate in the ILC3 compartment in mice. So we decided to set up experiments looking at this. And the question is around the idea, can these cells remember? So when they're activated, do they have any changes in their biology or in their long-term properties that allow them then to affect different types of biological effectors in time following this initial encounter with a pathogen? So the questions are listed here, and they surround the idea that when an ILC3 is activated in the context of infection or inflammation, that there might be changes that are durable and allow these cells to then be recalled with enhanced function. So we turn to a particular approach, which is very popular in way to study ILC3s in mice, and that's using a pathogen, a mouse pathogen called Citrobacter rodentium. There's an enteropathogen. It's an attaching and effacing bacterium, so it mimics E. coli infections in humans. And it's been shown by several groups that this type of pathogen activates innate and adaptive lymphocytes to produce IL-22, which is important for its clearance. So we wanted to know if Citrobacter actually modifies ILC3 biology in a durable fashion. And we thought about making an infection and then a secondary infection with Citrobacter. The problem was that Citrobacter, of course, activates ILC3s, but it also activates the adaptive immune system. So when you come back with the second infection, if you don't see anything on ILCs, it may be because the T cells and the B cells that make antibodies are able to protect the mouse. So we wanted to set up a system where we could activate just the ILC3s, but not activate the T cells and not to alter the microbiota, which is another aspect of Citrobacter infection. It causes a dysbiosis in the intestine of the mice that are infected. So we came up with this on-off infection model, and this was thanks to Gad Frankel, who's a collaborator and a Citrobacter expert in Imperial in London. And he had shown that certain antibiotics are very good at clearing Citrobacter infections. And based on his work, we chose an antibiotic, which was ciprofloxacin, to generate this type of a model. So the model is off. This is a mouse that's not been infected. On is the infection with Citrobacter. You activate the cells, you activate the immune system, and then you limit it. You limit it through antibiotics by treating the mice then with ciprofloxacin. That clears, the antibiotic clears the infection completely. And then you can take those mice and you can then rest them. And then you can come back and re-challenge them with another exposure to C-Renantium. And I won't show you the data. You can do this in a rag gamma mouse. Basically, you can clear that lethal Citrobacter infection in the rag gamma mouse that doesn't even have IL-22 by ciprofloxacin. And those mice are basically cured of that infection. And this model is a very robust way to activate the early steps of the immune response to Citrobacter without getting adaptive immunity to this pathogen. So here's what it looks like when you look at some data of infection. So these are fluorescent, bioluminescent Citrobacter that you can see in the intestine using whole body imaging. So mice after 3 days of infection with Citrobacter are colonized. They can then be cleared and reinfected a month later. When they're reinfected a month later, there's no sign of infection. So you don't visualize the bacteria. You can't find the bacteria in the feces of these animals. But there's a dramatic effect on the innate lymphoid cells that are present in the gut. And that's shown in the bottom right part of the slide, where you can see that the number and the production of IL-22 from these innate lymphoid cells is much higher in the re-challenge compared to the primary infection. So we call these ILCs that have now been trained, they're having memory features that are consistent then with a long-term reprogramming of these cells with enhanced function. So I won't show you all the different data, but rather summarize it in 2 slides. So these trained ILC3s have an impressive capacity to produce IL-22. They persist in the tissues for up to 4 months after they're activated primarily. They can be generated in rag mice, that's shown in the slide, with the ability of the rag mice that are primed with Citrobacter antibiotics and then re-challenged to control the infection. It's an IL-22 dependent process. It includes all the ILC subsets. And very interestingly, it can even be recalled in the second challenge by a diverse pathogen. So you can prime with Citrobacter and recall with Listeria, for example, and those ILC3s will be activated during the challenge with another pathogen. So these cells have a broad reactivity and a broad capacity to protect the barrier after they're initially activated by Citrobacter. One further property of these cells is that they're cell intrinsically able to generate this function. So if you transfer these cells using adoptive transfer, the trained ILC3s can protect mice against infection. And these are highly susceptible IL-22 deficient hosts, whereas naive ILCs are not. So these cells have a different property following that initial encounter. We wanted to understand what was the basis for that. We did several approaches to look at that using transcriptional analyses as well as functional ones. So I'll just summarize that data. Clearly, they have a different metabolism. These cells have an enhanced OXPHOS capacity with higher basal levels and higher reserves. It's clearly due to their mitochondrial function, and that is a major factor in the training of ILC3s and allowing them to be fit. In addition, these cells, as I mentioned, are able to make high levels of IL-22, but it's not only the production of cytokines, but as well their capacity to proliferate. That's extremely important. And here you see that in Ki67 on the bottom part of the slide, there are many more cycling cells in the mice that have been trained through Citrobacter exposure and antibiotic treatment when they're re-challenged. So that's the end of this story. Trained ILC3s promote intestinal defense. So clearly, like for T cells, when ILCs are activated, they have durable changes in their attributes and in their functions, and those can be beneficial to the host against certain types of pathogens and can be recalled by other types. So this is a very important way that the immune system learns to protect its barrier surfaces through experience. So to summarize the talk, I won't go through the slide completely, just rather highlight the idea that in situ ILC development may be operative in tissues, and that allows these cells to be generated where they're needed, when they're needed. So kind of on-demand approach to ILC differentiation. We still need to learn how these cells are regulated, how plasticity, which is another major factor in ILC biology, is regulated and important in some contexts. We have many observations for ILCs in different pathophysiologic conditions, but now we need to understand what are the key effectors of those ILCs in those disease settings. And once we can identify those, we can start thinking about ILCs as targets for immunomodulation in various human diseases, which I think will be very broad, not restricted to infection, chronic diseases, metabolic diseases, and many other diseases associated with aging. So I'll finish here, and I'd like to thank the entire team. It's a real privilege to work with such dedicated scientists. I have a really wonderful group here in Paris. I'd like to especially highlight Nicolas Serafini and Angelique Girard, who are really driving this work in the laboratory, as well as many other collaborators here at the Institute, in Philippe's lab, as well as international collaborators who played critical roles in this work by providing different mouse strains and expertise in these different areas. So I'll stop here, and I'll be happy to take questions.
Thanks, Jim. Yeah, I knew your lab have published so many wonderful paper about ILC3, but I didn't know how much you have done, and I today learned again these fantastic work. So Jim, we cannot get the question directly today. Okay, so let me give an announcement about this. So if anyone who wants to make a question to Jim, please do it. So through this Twitter. So please search for account Global IMMUNOTALK and find Twitter that said ask question for Dr. Jim Desant here. And please reply to the Twitter with your question and mention Global Immuno. So that's all, and thank you so much, Jim, again, and I hope to see you again, the audience, next week. So thank you very much.
Thank you very much, Kazuo. I hope to see you soon.
Yeah, me too.
Sleep well.